How do you measure the serum? Well, first, you need a sample from a patient that has antibodies in it. You need some sort of antigen of interest to compare those antibodies to, and then you need a method of detecting an interaction. However, if you're trying to discover novel causes of disease, which are associated with individual patient seromic profiles, you need an omics scale assay, something on the scale of the entire human genome. And so one way to do that would be to synthesize, purify, and print every single protein in the human body on a single glass slide. This is HuProt, the Human Proteome Microarray. It is the only protein microarray of its kind, which represents the majority of the proteins in the human body on a single glass slide. You take blood and you react it with the slide. You next come back with a detection antibody, which can be read on a laser microarray scanner. The intensity of the light will identify which unique autoantibodies are occurring in different patient populations. Even individuals that have the same autoimmune disease will still have exceptionally unique seromic profiles. However, what you will generally find is there is some unique autoantigen that they share. Where you start seeing things shared across a cohort of patients is typically where you start to see the things that might be associated with a causative mechanism of the disease. One of the main purposes of a tool like HuProt is it has full-length, 3D folded human proteins. These are really valuable for discovering whether or not relevant autoantibodies are occurring, which bind the actual folded structure of full human proteins, as well as target identification and other uses for discovering new types of protein-protein interactions and whether or not antibody drugs are hitting their intended targets. While the majority of an individual's autoantibody serome is generally thought to be harmless, there are some very clear and very profound cases where this is obviously impacting human health. The immune system can actually knock itself out, almost as if there was a genetic mutation. And what these drug-like autoantibodies do is they function as a phenocopy of these inborn errors of genetic immunity. In COVID-19, they've shown that for some people, why COVID-19 infection is dangerous at all is in part because of naturally occurring anti-interferon autoantibodies, which seem to impact and disrupt their natural immune response to COVID-19. And patients that have been given anti-PD-1, anti-CTLA-4. So these are the two cancer immunotherapies. And in some patients, you give them the drugs and it completely clears their tumor. Unfortunately, for a significant fraction, over half of the patients, they die anyways, and it's not really known why. Using our HuProt microarrays showed that the seromic profiles of these melanoma patients before they get cancer immunotherapy, they have a signature written in their autoantibodies, which seems to predict whether or not they will respond. It means there's something going on with the immune response prior to you giving the drug, which actually determines the outcome of this really important cancer immunotherapy. There's thought to be around 3,500 different surface proteins involved on the outside of human cells, and each and every single one of those is a potential case where perhaps an antibody to it could impact its function. If the immune system's allowed to knock out cytokines, what else might it be doing? Could it be knocking out cytokine receptors? Could it be binding and altering the function of GPCRs? These are all other things that the immune system might be unintentionally doing that interfere with our natural biologic processes and could contribute to disease.